Hello, everyone. It's Emma back again, and we are here to bring you another installment of the lecture series. This month, we have Dr. Karen Draths from Michigan State University, and without further ado, here she goes.
Hi, everybody. I'm really happy to be here today with the Science Squad. I think you guys are an impressive group of people who are, from what I'm told, you're very interested in science. So I'm glad to be here tonight and talk to you. Hopefully, you can all hear me. I don't know if there's a way to know if you can't hear me, so I'll just go ahead. So I'm in my second year at Michigan State as an assistant professor in the chemistry department. And as you can see from the title, I'm going to talk to you about how we make molecules in my lab using bacteria. That's my little cartoon of a little female bacterium there, and she's going to make some chemicals. All right, so organic chemists. I'm an organic chemist. So what does that mean? Well, if we look at the periodic table, organic chemistry is the chemistry that is involved with carbon. So you can see the element carbon is circled there. So organic chemists are usually interested in molecules that have carbon-carbon bonds. And organic chemistry is also the chemistry of life. So our bodies and the bodies and bacteria and animals and plants and things like that, that's all organic matter. So all that matter is carbon, nitrogen, phosphorus, sulfur, oxygen, all those green elements there. Here we go, on this side of the periodic table. All right, so as an organic chemist, sometimes we're interested in making molecules, and we would call that synthesis. We're synthesizing molecules. Sometimes we're interested in studying the physical properties of organic molecules. So there's all sorts of things that organic chemists might be interested in doing. I want to tell you about what we do, which I don't think you can see the top of the slide, but there we go. We do synthetic biology in my lab. So what is synthetic biology? Well, with synthetic biology, we use the tools of biology, specifically molecular biology, to make organic chemistry. So the biology part is what we're going to use as our tool, and we're going to use it to do synthesis. So generally with synthetic biology, we use bacteria as our host organism that's going to make these molecules. And so we, in my lab, oftentimes use the bacterium called Escherichia coli, which I know you guys have all seen that. That's E. coli. And, E. coli gets a bad rap in the news all the time when you hear people are getting very sick from E. coli poisoning. There are some bad strains of E. coli, but for the most part, E. coli, we all have E. coli inside our intestines, and E. coli is actually a pretty harmless organism for the most part, except those few bad ones. So in synthetic biology, what we're going to do is this is my little cartoon of DNA. And so this would be the big, in a bacterium, they'll have just one big piece of DNA. It's in a circle. This is not a good picture here, but it's usually in a circle. And what we do in synthetic biology is we can change the DNA inside the organism, and then that will change the mRNA that gets expressed. And of course, those mRNAs then get translated into enzymes. And so I've noted those as enzyme 1, 2, 3, and you can see this little enzyme 4 here too. So basically these enzymes do they catalyze reactions. So here, this red enzyme is converting molecule 1 to molecule 2, and the green one is molecule 2 to molecule 3, and so on and so forth. And so what we can do using synthetic biology is we can manipulate the DNA inside the cell, And in doing that, we change maybe how much of any enzyme is in there, or we can add new enzymes that aren't there, or we can do this, we can knock out an enzyme by making a mutation in the DNA. And these little circles here are plasmids. So these are very small pieces of DNA that are inside bacteria, and those are very easy for us to manipulate. And so as organic chemists, what we're interested in doing is we're interested in changing this DNA to change the enzymes so that we can make new pathways, so we can feed our bacterium a carbon source. Oftentimes that's sugar, something like glucose that we can get from corn. Okay, so we'll feed it a carbon source, and then our product will be made as it goes through the pathway. And one of the beauties of working with bacteria is frequently that product will be exported out of the cell, and then we can collect the product on the outside. All right, so that's synthetic biology. All right, so we're going to take a little medical time out here to talk about influenza. So I know you guys all know you maybe get your flu shots in the fall because you want to avoid getting influenza. So this is not the stomach flu. This is respiratory influenza that we're talking about. And in the United States last year, in 2017 and 18, it was considered a very bad flu season. About 80,000 people died in the United States. Usually those people are dying from pneumonia or things that they get subsequent to getting influenza. But in 1918, there was a Spanish flu. This is called a pandemic, where across the entire globe, millions and millions of people contracted an influenza. And in fact, 50 million people are estimated to have died in 1918, 17 and 18 from the Spanish flu. And actually, estimates are that it could have even been as many as 100 million people. Okay, so it turns out that approximately every 100 years, the Earth is hit with a strain of influenza that is both very transmissible from person to person, and it's also very deadly. And so you can see the last really bad pandemic we had was in 1918, about 100 years ago. So there's a lot of concern among, say, like the World Health Organization and other organizations that we are getting ready for another big influenza to hit us. Okay, so why am I talking about influenza? Well, let me tell you a little story here about sort of a relationship where we used our synthetic biology to help in the fight against influenza. So it starts by this molecule here, Tamiflu. Okay, so you might have heard your doctor or your parents talk about Tamiflu. It's a pharmaceutical that you can get. You can get a prescription from your doctor if you are diagnosed with respiratory influenza. And if you take Tamiflu when you have influenza, like the normal seasonal influenza, you know, it might make you feel a little better, a little faster, but for the most part, you're still going to feel pretty crummy, okay? So where Tamiflu is important is if we have a very lethal strain of influenza that's passed around, it's thought that Tamiflu is going to ameliorate the symptoms a little better, and perhaps the influenza won't be such a deadly strain if there's adequate supplies of Tamiflu for people. Okay, so Tamiflu was going to be developed by a pharmaceutical company called Roche in the late 1990s. And the problem that Roche had was that they couldn't get their hands on this molecule here. Okay, this is shikimic acid, and shikimic acid was the molecule that the pharmaceutical company Roche needed in order to make Tamiflu. So shikimic acid was their starting material. So I know you can see structurally how similar shikimic acid is. See that cyclohexane ring is very similar here over here. Anyway, so shikimic acid was not, there there weren't abundant supplies of shikimic acid. So where do you get shikimic acid? Well, it's a natural product, and you can get it from this thing here, which is called Chinese star anise, which if you go to the grocery store, you may see this. It's used in Chinese cooking, actually. It's a spice. And the problem with, so you can isolate shikimic acid from Chinese star anise. The problem is that Chinese star anise only grows in very limited geographical regions of Vietnam and China. And so it's not something that we can plant a bunch of here in the United States or in Europe and isolate. So Roche was wondering where they were going to get their shikimic acid. Okay, so we did a study and we looked at other types of plant sources that we might be able to get shikimic acid from. This one here, number 17, Illicium verum, that's the Chinese star anise. You can see that over 10% by weight of that little nut that you were looking at is shikimic acid. But you can see that's kind of a high percentage. Most of these leaves and stuff like this, ginkgo leaves, we have a lot of ginkgo trees on MSU's campus. They're about 5%, but still that's a very small amount of shikimic acid. And so we got into this. Oh, well, here's just a slide telling you that actually it was a big enough concern that this even made the New York Times, like, oh, where are we going to get problems ramping up the production of Tamiflu? Because there wasn't a big enough supply of shikimic acid. And so that's where we came in with our research. So let me show you here, forget these green arrows for a second. Okay, so I'm going to show you about shikimic acid biosynthesis in E. coli. Remember, we're back to our friend E. coli. So these are the enzymatic steps that E. coli uses, okay, to make phenylalanine, tyrosine, and tryptophan. Those are 3 amino acids that we use in our bodies, and animals and plants use to make protein. And so it turns out that humans don't synthesize these. We mammals all get our amino acids, or these amino acids at least, from our diet. But in E. coli and in plants, they make their own phenylalanine, tyrosine, and tryptophan, and they do it by this pathway. So what I'm showing you here is this, say this arrow F, okay? This arrow F is a gene that's in E. coli, and that gene encodes the protein, which we would also call arrow F. And arrow F will catalyze the combination of E4P and PEP to make this molecule here DAHP, okay? So that's how those steps go, all right? And if we go down the line, we see shikimic acid is one of the intermediates in this pathway. I got that red arrow showing you the shikimic acid. So it turns out that in E. coli, if you block, there are 2 genes that encode this enzyme here. If you make a mutation in those, so that X means that we're going to knock that out, what happens is the E. coli you'll feed it glucose or sugar, and the molecule, things will go down this pathway, the E. coli will grow, and then it can't convert shikimic acid to the next molecule here, shikimate 3-phosphate. So what happens is that the shikimic acid gets exported out of the cell into it's basically growing in water, like buffered water. So the shikimic acid gets exported out. So what we did was we amplified. These green arrows are showing you, remember I told you you could take these genes and you could put them on little plasmids, small pieces of DNA that are easy to work with. And so we manipulated our E. coli so that it had more of these green enzymes. It didn't need more of this one, okay? And so in doing that, now we're going to take as much of the glucose that we're feeding the cell as possible and send it down the pathway to try to make shikimic acid. Okay, so here's a plate, actually. This is an agar plate of E. coli bacteria, okay? So you can see all these little spots. And each one of these little spots contains like millions of cells in one spot, okay? And all of these spots on this plate should be identical, okay, because of the way we produced this plate. And so basically, this is just representing to you that because we've manipulated our E. coli, this is sort of the form that we get that E. coli. So we can take a plate like this that has lots of these little colonies, but we can just take one little colony off of that plate, and that cell, those groups of cells are going to be our catalyst, right? The catalyst is going to take glucose and it's going to make product. But the cool thing about our catalyst is that not only does it make our product, but it also makes more of itself because it's a living organism. And that's what's so fascinating for me about what it is that we do. So now that we have this plate with all these colonies, the next thing we're going to do is we're going to grow up lots of this, okay, in a fermenter. I'm going to show you a picture of that. Okay, so here's our fermenter, and this holds 2 liters. So think about like a big soda bottle. Okay, that's the volume of this this reactor here, and you can see there's lots of tubings going in and out. Okay, so we call this a batch top or a benchtop bioreactor, or some people will call it a fermenter. So you see all these tubings and stuff. That allows us to control how much oxygen we're sending in there. Of course, E. coli need oxygen to grow, just like we need oxygen to live. And so we give our E. coli lots of oxygen. We give it lots of sugar, glucose. That's its starting material. And then we can control the pH because we're producing shikimic acid as an acid. So because we're making lots of acid, we need to add lots of base in order to keep our pH neutral. And so that's our benchtop reactor that we use. And by using this, we can evaluate how much shikimic acid our little bacterium can make. Okay, so here is a graph of shikimic acid production. So if you look at these, so at time equals 0, so when we use our bioreactor, we put just a very small amount of cells in there. Oh, hey, hold on. There we go. Screwed up there. Hold on, I want to make sure Emma hasn't texted me. Okay, I know I'm way over time, but I'll go faster. Okay, so so these triangles are the cells growing up. So you can see over the first 18 hours, you're growing a lot of biomass, okay? So cells are growing, but we're not making a lot of product. And then after the cells stop growing, you can see that the black bars, which are the shikimic acid, grow in. And so now, from 18 hours to 60 hours, we make a lot of shikimic acid. In fact, we make 70 grams per liter of shikimic acid, which is a lot, okay? And these other bars here are byproducts that we want to get rid of. We can try to tweak our organism to get rid of those, but it's hard to completely get rid of them. So, hold on. Emma, are you here?
Yes, I am. You could try hitting the screen share button and taking it off and then putting it back on again.
Ah! What's that?
That's because you're open on the video window.
What should I do now, Emma?
Now just go back to the window with your slides.
The window with my slides.
Yep, just open up your slides again.
I just don't see it. Oh, there we go. There we go. Can you see it?
Yep, you're all good now.
Okay, sorry, I'm going long.
You're fine, you're fine.
Okay, all right, so the black bars then are the shikimic acid. All right. So when we do those in small scale, that's great. We can test it out and see how well it grows and see how much product it makes. But now this is a 30-liter fermenter that we have in our lab. This is my friend Yukari who works with me. And Yukari is showing you how, she would control the big fermenter. So when we grow our organism in this 30-liter fermenter, we can make over 2 kilograms of material at one time, which in a laboratory like ours, that's kind of impressive. So the beauty of what we've done, because we tested our organism in the small fermenter and then this big fermenter, is that those scientists at Roche who were concerned with how they were going to get their shikimic acid, we could just give them the information that they needed, and we could give them a plate, kind of like that little and we have all these cells growing in this water, but the cells have made the water basically black because it's made shikimic acid, and there's some other molecules that it's made too. And if we remove the cells, this is what we're left with, this black, dark material. It's like darker than coffee. But then if we want to purify the shikimic acid, we can go through a multi-step process, and this is the product. This is shikimic acid, and it's 98% pure, so there's a little bit of an impurity in there. But then the scientists at Roche, so MSU was able to take our technology and license it to Roche, and Roche has used our technology to produce shikimic acid to produce Tamiflu. Okay, so let's go through another story very quickly. Can we make a molecule that E. coli doesn't usually make? Shikimic acid is a molecule that E. coli knows how to make. Can we make a new molecule that E. coli doesn't make? And so I want to tell you about this molecule here, cis-cis-muconic acid. This is a molecule that can be converted into terephthalic acid is a monomer that's used to make plastic bottles and PET. Adipic acid can be used to make nylon, rope, cording, fibers, and actually it can be used to make carpets. And so we had interest in whether or not we could make this molecule using our synthetic biology. And so we're going back to that same pathway that I showed you before where we made shikimic acid, but now we block this step. That's what these bars mean. And then what we do is we take 3 genes that don't exist in E. coli, ARO-Z, ARO-Y, and CAT-A. Those aren't ever found in E. coli. And we took the genes from other organisms and we put them in E. coli and we expressed them. And now our organism should make cis-cis-muconic acid. And so this graph basically shows you that. So again, the diamonds are cell growth, and then the blue bars are the cis-muconic acid. So we created an organism that could, so that was our E. coli, that could take glucose and convert it to cis-cis-muconic acid, and then it can be reduced to adipic acid, which can be used to make nylon. So up till now, I've been telling you about all these things that we've done where we feed our bugs glucose. And the thing about that is, so glucose and sugar, those are over 40% of the corn crop that we grow in the United States is being used to make bioethanol. And while that might sound like a good idea, we need a lot of commodity chemicals in our country and in globally. And so to be using things that we get food from to make chemicals probably isn't a good idea, because we already have starving people on this, on the earth, and populations are only growing. And so this is kind of problematic. And so we're turning to a different type of, we have a different idea. And so what I want to show you is these green splotches on our map are places where we are currently drilling for what's called shale gas. So these green spaces are where there is fracking, hydrolytic fracking that's going on. And so what we're doing now is we're tapping down, deep down underground, and we're horizontally drilling. And from that, we're getting shale gas, okay? And shale gas is another form of carbon. Hold on one second, I'm gonna go forward. So shale gas gives us this methane, okay? Methane, which is also natural gas. Okay, so this is our little thing of hydrolytic or horizontal drilling and fracking. Okay, that's where we're getting all this methane. In fact, our natural gas prices are very, very low these days because the United States is producing so much natural gas. Okay, and I want to show you another picture. You see these green and blue off the coasts, the continental coasts, okay? These indicate this is where there's lots of methane underneath the ocean, okay? And there's a picture here. This is methane hydrates, okay? It's basically like ice crystals of methane, and they're deep down underneath the ocean floors. And currently, there is
You seem to have frozen a little bit there, Karen. We can't see or hear you anymore. I'm actually going to use this time to see if the internet reloads, to say that if anybody has any questions for either Karen or us, they can email us at the Science Squad or go to our Facebook page and message us with any questions. You can also comment right now in the broadcast, and we will see if we can figure this out.